So hello students and welcome back again to yet another video tutorial by Genome Biotech. In this video tutorial, we will be talking about site-specific recombination. What do you mean by site-specific recombination? How does site-specific recombination takes place in the case of phage integrating its genomic DNA into the bacterial cell? And what is this site-specific recombination? So as you all know that a region of extensive homology is not a prerequisite for the recombination to take place. We never require a very great amount of homology in between the nucleotide molecules so that the recombination should take place. Uh, Recombination can also take place in between two different DNA sequences that shares very less homology. Okay, so such type of recombination has been shown to take place in between the lambda phage POP- site and the bacterial genome BOB- site. This causes the integration of the lambda phage DNA into the bacterial chromosome. If you look here in the diagram that I'm showing it to you, here is the bacterial chromosome. See, okay, there is a bacterial chromosome which is present. To this bacterial chromosome, the phage binds to the bacterium. When the phage binds to the bacterium, the phage has the ability to insert the genetic material into the bacterium. Okay, it would be inserting the its own genetic material into the bacterium. Now taking the phage with another color, supposedly red color, and this is the genetic material of the phage that has been incorporated. Okay, this genetic material of the phage can have two different fates. Okay, the phage can undergo two different types of life cycle. One, it could undergo lytic, another one, it could undergo lysogenic life cycle. Okay, so it can have two different phases, which are the two different outputs: lytic cycle, and another one is lysogenic cycle. In the case of lysogenic cycle, what would have happened? In the case of lysogenic cycle, the phage DNA would have been incorporated into the bacterial chromosomal DNA. Supposingly, the phage DNA gets incorporated into the bacterial chromosomal DNA, then such type of phage DNA will be referred to as the prophage. The prophage. Prophage, as you know, refers to an inactivated phage. This prophage refers to an inactivated phage, inactivated phage produced produced as a result of incorporation, produced as a result of incorporation of phage DNA, incorporation of phage DNA into the bacterial chromosome, into the bacterial chromosome. This is how the integration is going to be performed. This is the manner how the integration is going to be performed, and in what manner the integration is going to take place. Now, for this integration to take place, the phage and the bacterial chromosome has a common region that is referred to as the O region. The O, which is referred to as the common site which is present between between the lambda phage DNA and the bacterial chromosome. So this is the lambda phage, and such type of integration will take place only in the case of the lysogenic cycle, because there is no integration in the case of lytic cycle. Integration takes place only when lysogenic cycle takes place. We would be clearing the lytic cycle and the lysogenic cycle in a great depth when we would be talking about the operon model or the lambda phage operon, okay, that we would be looking forward in the lectures to come. Okay, so a region of extensive homology is not a prerequisite for the recombination to take place. The process can also be initiated between two DNA molecules when they have a very short sequence in common. So when the two DNA molecules have a very short sequence in common, then also the process of recombination can take place. And such type of recombination is commonly referred to as site-specific recombination. here is referred to as site-specific recombination, where the phage DNA can integrate into the chromosome at a particular site. Integration of bacterial phage DNA into the E. coli genome involves a site-specific recombination. That is, it is going to take place at a particular site. The sites are referred to as the add site. Add site refers to attachment site. So there are two different add sites. One is present, one add site is present in the case of phage, lambda phage. Another add site is present in the case of bacteria. This is present in the case of bacterial genome, that is E. coli. It is present in the case of E. coli. Okay. Next, the lambda phage. So one attachment site is present on the lambda genome and another one is present on the E. coli chromosomes. Okay. Both these attachment sites have a common sequence present within itself, and that sequence is referred to as a 15 base pair sequence called as the BOB dash and the POP dash. So there is a central region which is referred to as the O region, which is common in both bacteria as well as in the case of phage. So this centrally present O region is common in both of these organisms. So this is that particular O region. Now the recombination is going to take place. The sequence O is common in the attachment B and the attachment P site. It is called as the core sequence. This site which is present is referred to as the core sequence, and the recombination only occurs in this particular sequence. It is only in this particular sequence that recombination is going to take place. The recombination event is actually being catalyzed by a specialized type 1 topoisomerase, which is commonly referred to as an integrase. So there is going to be an enzyme that is referred to as lambda integrase that is going to catalyze the integration of the phage DNA into the bacterial chromosomes, so that so that the absolute integration can effectively take place. The enzyme makes a staggered double-stranded cut at equivalent positions because it is integrase the topoisomerase. Integrase enzyme is present, and this integrase which is present serve as a topoisomerase, as it is a topoisomerase 1. So this topoisomerase 1 makes a cut in the DNA molecule. It is going to make a cut here and it is going to make a cut here also. Okay. And then the integration is going to take place in such a manner that there would be BOP dash. Now, how BOP dash could be produced? It could be produced like this: BOP dash. And then the entire genome will be made, and then POB dash. From here, it would be POB dash. And then the integration will be made in this particular manner. Okay, if you are not getting it, please look here again. Please do look here, that how the integration is going to take place. This would take place BOP dash. BOP dash. P dash means the O is going to attack at this particular location. How is the attack going to be mediated? I'm just going to show it to you with a yellow color. See this. This is how the attack is going to be mediated. Okay. And from here, when this bonding will take place, what would be the sequence produced? BOP dash. See this? The sequence of the genes that would be produced will be BOP dash. Another one, BOP dash. And when the circle will be completed, then it would be P dash OB dash. P dash OB dash. Or BOP dash, BOP dash, and POB dash. These are the two sequences that will be made. Okay. The two short single stranded overhangs are then exchanged between the DNA molecules. It will produce a junction that is commonly referred to as holiday junction, which migrates a few base pair along the heteroduplex before it is being cleaved. The holiday junction is going to be produced. The holiday junction is going to be migrated. The two single stranded DNA molecule will form overhangs and are then exchanged between the DNA molecules. It will lead to the production of holiday junction. Once the holiday junction is being produced, there is a heteroduplex which is being formed. The heteroduplex is being cleaved, and the resolution of the holiday junction is going to take place in such a way that the lambda genome becomes integrated into the E. coli genome. Ultimately, the lambda genome will get integrated into the E. coli genome, will form the double stranded DNA molecule. So, this is how the integration is going to take place. Is this clear to everyone? I'm going to show you the animation and we are going to discuss it with the animation also. Okay, so no doubt about this, how the integration is going to take place. Now, let us have a view upon the animation that is being given here. So, I would be showing you that animation. Directly moving to the animation part. Now, so what we have to discuss here is about conservative site specific recombination that we have already discussed. We are going to visualize the animation. So please do have a view that how the two DNA molecules can get integrated, how the lambda phage can integrate its DNA into the E. coli chromosomal DNA. Please look.
DNA sequences between different DNA molecules. Conservative site-specific recombination, or CSSR, is recombination between two defined sequence elements called site-specific recombination sites. There are two families of proteins responsible for site-specific recombination: the serine recombinases
And the
and the
tyrosine recombinases are the family of proteins that are going to cause the recombination to take place. Now, how the serine and the tyrosine recombinases are going to work, that we have to understand. So these are the two family of proteins. One is referred to as the serine recombinases. Another one is referred to as the tyrosine recombinases. Now, how recombination is going to take place, in what manner the recombination is going to take place, that we have to see. Here you can find that the two DNA sequences shares the same direct repeat sequences, so they can be effectively cleaved by the same endonucleases, and once cleaved and once they would be cleaved, they could be exchanged in between the two DNA molecules, and once the exchange is going to take place, they will form new different types of combinations. So moving forward, how does this combination is going to be formed?
tyrosine recombinases. When you have completed this exercise, you should understand the mechanism of site-specific recombination by the serine recombinases. Understand the mechanism of site-specific recombination by the tyrosine recombinases. And understand the possible outcomes of conservative site-specific recombination.
So there are three things that we have to learn. We will have to understand what is the mechanism which is being used by the serine recombinases during the process of recombination. We must understand what are tyrosine recombinases, how does tyrosine recombinases act, and the last and the most important point, we must understand about the possibilities of conservative site specific recombination. What is this conservative site specific recombination, and how does the conservative site specific recombination actually acts in the cell? So please to have a view.
For the serine recombinases, the side chain of a serine residue within the active site attacks a specific phosphodiester
Okay,
bond in the recombination site.
this.
This reaction introduces a single-stranded break in the DNA and simultaneously generates a covalent linkage between the serine and a phosphate at the cleavage site of the DNA.
Now the main enzyme that is referred to as the topoisomerase. Okay, that is referred to as the type 1 topoisomerase, because ligase is a type 1 topoisomerase. Already it has been told to you. The ligase enzyme which catalyzes the site specific recombination, which catalyzes catalyzes the site specific recombination to occur, is a type 1. Oh, sorry, it is not ligase. The topoisomerase enzyme, that is integrase. The integrase enzyme which catalyzes the site specific recombination to occur is a type 1 topoisomerase. Topoisomerase polymerase enzyme. So will firstly cleave the phosphodiester bonds, and again reforms the same after the supercoiling has been removed. Supercoiling has been removed. So once the supercoiling has been removed, the phosphodiester bonds can be again produced. So such would be the activity of the integrase enzyme. So here the integrase enzyme, which is actually a serine recombinase in this case, is going to cleave the phosphodiester backbone. And once the phosphodiester backbone is being cleaved, the enzyme is going to release the civil, it is going to release the supercoiling from the DNA molecule, and then again reform the phosphodiester linkages. So moving forward.
Site-specific recombination always occurs between two recombination sites. Therefore, during recombination, 4 single strands of DNA, 2 from each recombination site, must be cleaved and rejoined. The serine recombinases cleave all 4 strands prior to strand exchange.
Now, the process of recombination always involves two different DNA molecules. Okay, so this is a homologous chromosome. This is one part of the chromosome. Supposingly, this is the lambda phage genomic DNA. Then the other genomic DNA will be that of the E. coli. For example, this is lambda phage genomic DNA. Okay, genomic DNA. Now, this genomic DNA has to be integrated with the E. coli genomic DNA. It has to be integrated with the E. coli genomic DNA. Genomic DNA. Now, how this integration is going to take place? This integration is going to take place when the cleavage of the phosphodiester backbone is going to take place. Here, the cleavage of the phosphodiester backbone has been taking place. Once the cleavage of the phosphodiester backbone has taken place, then the exchange of the genetic information could take place. Once the exchange of the genetic information could take place, then the ligation will take place. So, moving forward.
With one recombinase protein molecule acting on each strand.
Okay, so there is a single recombinase protein that is going to act over each and every strand, and this would be the recombinase protein that would be acting on both the strands of the DNA molecule. Moving forward.
The covalent protein DNA intermediate conserves the energy of the cleaved phosphodiester bond. As a result, the DNA strands can be rejoined simply by reversing the cleavage reaction.
Now, the DNA bonds could be just joined by reversing the cleavage reaction. Now, the cleavage reactions can be reverted back and the molecules could be joined just with regard to each other. This is how the molecules are being made to join.
Site-specific recombination is called conservative because every DNA bond that is broken during the cleavage reaction is resealed by the recombinase without any input of external energy.
Now, because this is a topoisomerase, as I have already indicated that the enzyme which is responsible for the integration or for the site-specific recombination is a type 1 topoisomerase. Integrase is a type 1 topoisomerase. So will not require any external input of energy. Firstly, it will cleave the phosphodiester bond in the pre-existing strand that was present in between the blue and the blue strand, conserve the energy, use that energy in order to remove the negative or the positive supercoiling, and then introduce the ligation reaction to take place, will join the phosphodiester bond, uh, causing the ligation in such a manner that the genes have been exchanged in between the two target DNA molecules. Look here.
The mechanism of action of the tyrosine recombinases is very similar to that of the serine recombinases, except a tyrosine side chain is involved in the cleavage reaction. Rather,
Now, see, tyrosine recombinases are going to function exactly that with the serine recombinases, except the only exception being the fact that in the case of tyrosine recombinases, whenever you are going to talk about tyrosine recombinases, you have to stick here that tyrosine recombinases will use the tyrosine amino acid that will be cleaving the phosphodiester backbone. Once the phosphodiester backbone is being broken down, tyrosine recombinases has formed uh, itself a ester bond with the phosphate molecule. It will release the supercoiling, will again rejoin the phosphodiester bonds in between the
Other than
molecules.
a serine side chain.
Next point.
Also, in contrast to the serine recombinases, the tyrosine recombinases cleave and rejoin two DNA strands first, generating a holiday junction.
This will generate
The
a
other two
holiday.
tyrosine recombinases then cleave and rejoin the other two DNA strands, resolving the holiday junction.
Now you can look here. There are two holiday junctions which have been produced. Okay, because this is tyrosine recombinase. Tyrosine recombinase just differs from the serine recombinases in which there was a single holiday junction being produced. Here there are two holiday junctions which are being produced because it is going to cleave the DNA molecule in such a manner that the two homologous recombination type of molecules are being made. Okay, here see this, these are the two holiday junctions, and now the genes will be exchanged. How? Look here, see this is the new variations which are going to be produced. So tyrosine recombinases introduce much more variation as compared to the serine recombinases. So here I can conclude one thing, and the thing that has to be concluded is that the recombination involving the tyrosine TYR recombinases recombinases will produce more number of recombinant molecules as compared to the serine recombinases. Okay, as it will involve the formation of two holiday junctions. Two holiday junctions. So there are two holiday junctions which are being produced. So it will involve the formation of two holiday junctions, thus will produce different types of combinations. More amount of combination will be produced in this particular event. Moving forward.
Each recombination site is organized as a pair of recombinase recognition sites, positioned symmetrically. These sequences flank a central short asymmetric sequence where cleavage and rejoining occur, called the crossover region.
Now, just you compare this particular sequence with what I have already told you from the Pathfinder. You just compare this sequence with this. There is a centrally aligned region which is similar in between both the DNA molecule, and then there are short direct repeats which are present. These repeats will be the site where restriction endonuclease is going to work, where the endonuclease are going to be in action. Moving backward to the animation. Look here.
If the recombination sites are on different circular DNA molecules,
See, this is a simple recombination that is going to be observed here. You just remember this as POP dash, POP dash, that is present in the case of the lambda phage genome. Let this be a lambda phage genome, and the other genome be that of the E. coli. Okay, this is the lambda phage genome, and this is BOB dash, the type of the genetic material that is present in the case of the E. coli. So you can find it, this is E. coli, bacterium. Now moving forward.
the result of site-specific recombination is in
Now see how the results of the site-specific recombinations are going to be produced. I will not disturb it until and unless the site-specific recombination is over. Please do have a keen view.
insertion of one of the molecules into the other.
Now, this is what we have gotten. The lambda phage genomic DNA has now been integrated into the bacterial chromosome. So, what you are going to find here is that this is the lambda phage genomic DNA, which has been integrated here into the bacterial chromosome. And this is the entire bacterial chromosome that is going to be produced. Okay. This would be that particular bacterial chromosome that has been formed after the integration has taken place. So, you can find that the molecules that would be produced out of it will be in the form of BOP dash and POB dash. So this would be BOP dash and POB dash. BOP dash and POB dash. This would be the lambda fast genome. Just comparing it with the pathfinders. Remember what I wrote: BOP dash and POB dash. BOP dash and POB dash. Moving backward to our animation. See this? Okay. BOP dash and POB dash. Moving forward.
Because the crossover region is asymmetric, a given recombination site always has a defined polarity. If both recombination sites are on the same DNA molecule, they can be a direct repeat with respect to one another or inverted with respect to one another.
Now, there are two different types of repeat sequences that could be there. The repeating sequences can form either a short direct repeat or they can form an inverted repeat. What do you mean by short direct repeat? Short direct repeat is a type of repeat in which the same sequences are being repeated again and again. Then it is referred to as a short direct repeat. You just have to look here. This is the repeat sequences that have been repeated here also, and this is referred to as a short direct repeat. The same sequence is going to be repeated. Okay, the same sequence is being repeated in the genome, then it is referred to as the short direct repeat. When the same sequence is being repeated across the genome. Okay, this is very important. When the same sequence is being repeated across the genome, then it is referred to as the short direct repeat sequences. Okay, now you look here, what happens in the case of inverted repeat sequences. In the case of inverted repeat sequences, you can find although the same sequence is being repeated but in an inverted manner. When the same sequence is being repeated in the genome but in an inverted manner. So there is going to be a repetition that is going to take place, but the sequence is going to be repeated in an inverted manner. Then it is referred to as an inverted repetition. Okay, so what we get here is that there is a repetition that is going to be produced, but this repetition is going to be inverted by an angle of 180 degree. Okay, there is a repetition that has been inverted by 180 degree. So I can just rotate this out. Sorry. Okay, and just repeat it as so this angle has been repeated by 180 degree. Okay, so this is referred to as the inverted repeat DNA sequences formed as a result of site specific recombination. Now these will be the sites where the recombinases are going to act. Now how the recombinases are going to act, that we must visualize. Please look at the outcome that would be produced.
Recombination between a pair of recombination sites organized as direct repeats will result in deletion of the DNA segment between the two sites.
Now, this is the most important thing. So, if there are two direct repeating sequences, then the process of recombination between the two direct repeat sequences, there are two direct repeat sequences. First, here, another direct repeat sequence is here. Then the two direct repeat sequences will undergo recombination and will lead to the formation of such type of recombinases. You can look here. There is this type of a recombinase. Oh, sorry, there is this type of a recombinase that will be produced. This would be a type of a recombinase that would be produced. You can look here. This is the type of a recombinase that has been produced. Okay. So I'm just clearing the drawing here. Now, this is because there has been an inverted sequence that has been produced. Because of those inverted, because of those two short direct repeat, there is a recombination, and then the DNA along with the gene has been expelled out. Okay, so because of the short direct repeat, the DNA along with the gene has been expelled out. The short direct repeat, the short direct repeat that is flanking, that is flanking a gene of interest will be cleaved through homologous recombination in such a manner that the gene of interest will be expelled out. Okay, this is the gene of interest that is going to be expelled out. This is what we have to study here. Now, to understand this particular point, please do look here. Please do look here at the Pathfinder, the new edition of Pathfinder. We must look at a system that is referred to as a Krelok system. Now, Krelok system, see, this is what here has been given. See, integration of the lambda fast. See, integration create hybrid version of the attachment site, which is referred to as at L. Now, this is a hybrid attachment site that is going to be produced at L with the structure BOP dash, and at R will be produced, and the at R will have a structure that is referred to as POB dash. So there are two sites which are going to be produced. One is referred to as BOP dash, another one would be referred to as POB dash. POB dash, that is at R, POB dash. These are the two attachment sites that would be produced. Excision of the lambda phage require another recombination between the at R and at L, and then excision will be caused in association with the integration host factor and the XIS protein. XIS protein refers to as the exogenous protein that is found in the case of the bacterial cell. If the lysogenic inversion has to take place, what do you mean by the phenomena of lysogenic inversion? There is a phenomena which is called as lysogenic inversion. Lysogenic inversion. Lysogenic inversion is the phenomena when the lysogene gets converted into a phage. When the lysogene gets converted, when the lysogene transforms itself into phage, transforms, trans, when the lysogene transforms itself into phage, transforms itself into a phage and began to perform lytic cycle, and began to perform lytic cycle began to perform lytic cycle to perform lytic cycle. The process is commonly called as lysogenic inversion. Okay, this would be referred to as lysogenic inversion. Now, in the case of Krelok system, why I'm talking about the Krelok system, because I'm going to talk to you that how the recombination is going to take place. Now, Krelok system is actually a type of tyrosine recombination system. In this, the tyrosine is going to be recombined. The tyrosine recombinase are going to function. Now, which would be the most important tyrosine recombinase? It belongs to an integrase family, as there was an enzyme integrase which was responsible for the integration of lambda phage into the chromosomal DNA. So there is an enzyme which is present for the activity of the Krelok's P system. Okay, this is a type of system in which a long P site 34 base pair long consists of 13 base pair inverted repeat sequences. There is a site that is referred to as LOX P site. And this LOX P site consists of 13 base pair inverted sequence, and the centrally present sequence. The centrally present sequence is of 8 base pair. You can find here, this is the centrally present sequence. The centrally present sequence is of 8 base pair. It is already written, and there are sequences flanking this 8 base pair region. Which are those sequences? Those are the inverted repeats. You just have to look. These are the repeating sequences that you can find. TATA, TATT, GAAGCATAT. Okay, so you can find that these are the same sequences that have been inverted. Just they have been inverted and they have been joined. So you can see this. This would be 5 prime. Let this be 5 prime, and this is 3 prime. Now you just look here, which would be the 5 prime sequence. There would be a 5 prime sequence on the complementary end. This would be the 5 prime sequence. Okay, and this would be the 3 prime sequence. You just look here between these two sequences. This is 5 prime, that one was 3 prime. ATA, ATA, CTCTC, CTTC, TATA, TATA. So these are the two different sequences which are present. So you can just match them up. This is the sequence in this direction. This is the sequence in this direction. So this is just an inverted repeat sequence and nothing else. Now, what is going to happen? The Cree recombinase is going to work. Cree recombinase is actually a integrase. It is an integrase that will catalyze the integration to take place. The orientation and location of the LOX P is going to determine whether which type of recombination is going to take place. Now you just have to memorize these things. See, if the cleavage is going to take place and at this particular location, inverted repeat, supposingly there is a cleavage that takes place here. The similar type of cleavage is also going to take place here. Okay, at this particular sequence, after these sequences, this will produce a complementary sequence. This will produce a complementary sequence. You can see that the two DNA molecules will be complementary. Because the two DNA molecules will be complementary, so they will form a circular structure. This is what is seen here. This is a LOX P. LOX P. These two LOX P will be cleaved with the help of restriction endonuclease. The two restriction endonuclease are going to cleave this. When the two restriction endonuclease are going to cleave it, then it will form complementary sequences. When the complementary sequences will be produced, it will form a circular structure. And once the circular structure is going to be produced, then this part, this central part, the central gene will be completely lost. For example, this is the gene X. See this, the gene X, which is being encoded by the green color here, is completely being lost. This gene X has been lost. Gene X made to lost. This is the gene X which is going to be lost. Anything, any doubt? So whenever the LOX sequences or the inverted repeat sequences just flanks a particular gene of interest, then the gene of interest could be lost by the process of recombination. This is referred to as the Cree-Lox recombination procedure. Any doubt, any problem to any student, you just ask. You just ask. Any doubt, any problem, any student, please ask. Hello, any doubt, any problem, you just write here also, and I will answer. No, this is very clear na. Whenever a gene of interest is being flanked, whenever a gene of interest is being flanked by two LOX P sites, the gene is being lost. Now the LOX P has two opposite orientation. Two opposite orientation. When the LOX P has two opposite orientation and the cleavage is going to be made, the LOX P has two opposite orientation, one in this direction and another one in this direction, and the cleavage is going to be made. Once the cleavage is going to be made in this particular arena, it will cause inversion of the gene X. Gene X is going to be inverted. Gene X is going to be inverted. There is an inversion that is going to take place. So if the two locks are in opposite direction, gene X will undergo inversion because they will be cleaved in such a manner that the gene X will undergo a 180-degree turn, and then the same gene will be joined over the DNA molecule. Now, when the two locks sites are present on the two homologous chromosomes, then they will undergo recombination. How they will undergo recombination? This would be the site where the recombination can take place. It would be cleaved like this. Another locks will also be cleaved in the similar manner. It would be also cleaved in a similar manner. The two DNA will be exchanged at this particular site. So this DNA will move above and this DNA will move down. This DNA will move above and this DNA will be made to move down. See this? The light brown color DNA has been made to move above and the black DNA has been made to move down. This phenomena is commonly referred to as the translocation. Translocation refers to the exchange of the genetic material. The exchange of DNA fragments have taken place. Exchanged of the dsDNA of the dsDNA has occurred. Exchange of the dsDNA has occurred. Sorry, just writing in a close manner. dsDNA has occurred. Probably this is visible to you all now. Exchange of the dsDNA has occurred. If not, I can take another color. Okay, I would be asking you certain questions from this particular part. There are 2 3 questions that have been asked, and we would be discussing that particular question in this lecture only. Okay, so the Cree and LOX recombination system, just changing the color. So the Cree LOX P system is a system which has been applied in a number of different experimental ways, including the site specific recombination system. So this is a system that has been used to knock out certain genes, okay, to study certain genes. So it is a system which has been used to study the specific features of to study the pattern of inheritance of genes, to study the pattern of inheritance of particular gene, pattern of inheritance of a particular gene, how the pattern of inheritance of a particular gene is going to take place. It is used to study the to make temporal knockouts of the gene, to make knockouts of a gene, knockouts of the gene, of the gene, and to study about its functionality, and to study about its functionality. So what would be its function that we will have to study? Okay, so these are the two systems for which it has been used. Now please do look here, that how it has been used, in what manner it has been used. The condition activated an inactivation of transgene and the deletion of unwanted marker genes. So it will cause the degradation of certain genes and will lead to the expression of unwanted marker genes. The flap fat unwanted marker genes, the FLP frat system is similar to the Cree-Lock system. Now, this is the most advanced system which has been used in molecular biology to study the genomic integrity and what is the function of a particular gene, how that gene is going to be functional in a cell. We use another system that is referred to as the FLP flap frat system. Okay, it involves a recombinase that is referred to as the FLP recombinase derived from the yeast cell Saccharomyces cerevisiae and the FLP recombination target, just like Cree and LOX. So the function of Cree will be performed here by the FLP recombinase. This is going to perform the function of Cree, and the function of lox p will be performed by the target site. This will perform the function of lox p. Rest all will remain same. A genomic DNA kept in between the two FRT sequence will be lost. See this? A genomic DNA which has two FRT sequence. I'm just keeping it in between the two FRT sequences. And now, in what manner the FRT sequences would be kept? Okay, I will keep it in blue color. So, supposingly, if this is FRT sequence, if this is the first FRT sequence, FRT, there is another FRT sequence in the same manner that has been kept just by short direct repeat sequences. See this? Then what is going to happen? Then the recombination is going to take place and this particular gene will be lost. It would be lost. It will undergo deletion as the gene has undergone deletion there. So such type of system which is being produced is referred to as the fret flap system of recombination, which is also referred to as FLP FRT system. FRT system. This is the system that is going to be produced. Any doubt, any problem, you just ask. Okay, this is the system that is going to be produced. Wait for a second. We are going to move forward and look at the questions which have been asked in CSR net entrance examination and in other entrance examination. FRT is a 34 base pair long sequence. It has two inverted repeat sequences of 13 base pair, and these are being separated by an asymmetrical sequence of 8 base pair. So there are 13 base pair sequence here, 13 base pair sequence here, and there is an 8 base pair sequence. These will be referred to as the FRT, and FRT. And here there would be the recombinase that would be referred to as, these are the sites. These are the two different sites. The recombinase will be FRT recombinase, FRT recombinase, which is going to be functional. Binase. That will function here. Okay, any doubt? Or will function at this particular sequence. Will cause the cleavage, will cause cleavage and recombination. Cleavage and recombination. It will cause the cleavage and recombination to take place. Now, moving backward to the animated version of the lecture and completing that animation, then we will look at the questions. Okay, have some patience. The questions will be coming in front of you.
In contrast, recombination between a pair of inverted recombination sites will invert the DNA segment.
Now see this. A recombination between inverted site will cause the inversion of the DNA sequence. This is what they have told you. See, in contrast, recombination between a pair of inverted recombination site will cause the inversion of the DNA sequence. This is what I have told you, that if the recombination is going to take place in between the two inverted sites, the two inverted locks site, or between the two inverted FRT sequences, then the gene present in between them will be inverted. This has happened in the case of inversion. So in the next example, our animation is talking about this particular scenario, this particular condition in which there are two locks sites and the gene will be inverted in these locks site. Any doubt, any problem, you just ask me. Okay. Use the chat button, write your message, and I will be answering it to you. Is this clear to you, everyone? Is this clear to everyone? Now, moving forward.
Mice are often used for studying human diseases, such as
Now,
cancer.
we must look at those questions which have been asked in CSR and GRE entrance examination. These questions are really good. Now you have three different case scenario in front of you, that how the recombination is going to take place, in what manner the recombination will take place. Just to have a short recap, just 1 minute, listen to me. That recombination was conservative site specific recombination was a process in which the lambda phage was able to integrate its DNA into the bacterial chromosome. We have already seen the same process. And then this type of process we have seen, this method has also been used in the case of Cree locks in the form of FRT, and the FRT recombinases, that is FLP and FRT system. Okay, and this ensures that we can make a temporal gene knockout or a permanent gene knockout, as well as invert a particular gene and then find out what would be the activity produced by that particular gene, or what are the effect produced when a particular gene is being deleted from the cell. And in this case, when the two locks P or the FRT sequences are on the two homologous chromosomes, the reciprocal exchange of the genetic material is going to take place in between the two, referred to as translocation. Now, move it out and solve the different questions which have been asked in your entrance examinations. So, look forward, and I'm repeating the slide. Please do listen very carefully.
Mice are often used for studying human diseases, such as cancer. It is often useful to be able to knock out a specific gene within the mouse genome to study the effects of that gene in the development of a given disease.
Now, we always know that mice have been extensively used in order to study cancer. It is often useful to be able to knock out a particular gene. So they are talking about a knockout of a particular gene. So knockout of a particular gene, that means silencing a particular gene is of specific importance for the mouse genome to be studied, to study the effect of the gene in the development of a given disease. Now, please have a look.
You have identified a gene found in the genomes of both humans and mice. You believe that the protein product of this gene helps to prevent cancer in both mice and humans. How can you study the effects of this gene?
Now you are in a particular dilemma. Do the protein products will help in the fight against cancer? Do protein product of this particular gene protein product will protein product of this particular gene will help to prevent cancer, and how or not the cancer will be prevented or the cancer won't be prevented, that you have in your mind. You just have to look at that particular case scenario. So let us have the first question. There is a gene, there is a protein. You have to just understand whether the protein product of that gene will be looking to prevent cancer or not.
Site-specific recombination is a useful tool for such studies. CRE recombinase is a well-characterized tyrosine recombinase. CRE recombinases cleave at recombinase sites called LOX sites.
Now, for such type of a case scenario, in order to produce a particular knockout gene, knockout gene production involves the use of a particular recombinases, that is referred to as the Cree recombinases or CRE recombinases. CRE recombinases are one of the most effective tool which act upon a particular site, and that site where they are going to act is referred to as the LOX recombinase site. Now, how they are going to act, please have a look here.
Question 1. Which of these experimental techniques could be used to flank the target gene in the genome of a mouse embryonic stem cell with inserted LOX recombination sites?
Now see, which of these experimental technique can be effectively used in such a manner that it could be used to flank the target gene in the genome. Now, which technique can be used to flank the target gene in the genome of a mouse embryonic stem cell with inserted LOX recombination site? Now, what they are trying to say to you is that to flank the gene, you just have to delete a particular gene. Now, which of the following techniques can be used in to delete a particular gene. When you talk about DNA footprinting assay, it is used to identify the site where protein binds over the DNA, the site of protein attachment over the DNA, of protein attachment over the DNA, attachment to DNA. This site is being determined with the help of a protein that is referred to as DNA footprinting assay. PCR, as you know, it is a gene amplification technique. It is a simple gene amplification technique. Okay. And the last, homologous recombination can be used in order to flank the target gene and then inhibit a particular gene of interest. Okay, so we can use
Correct.
the homologous recombination. This forms the correct answer. Now moving back to the next question. So this type of method of recombination can be used to flank the target gene in the genome of a mouse and to study that particular gene.
Question 2.
Now, before we move to the question number two, I must delete the annotations. Now, moving to the question number two.
You wish to delete the target gene. Where should the lock sites be inserted? Be sure to set the orientation of the lock sites.
Now, we just have to delete a particular target gene from the chromosomal DNA. Now, if you have to delete a particular target gene from the chromosomal DNA, then what has to be done? The target gene has to be deleted. Now, in which manner the target gene has to be deleted? For deleting a particular target gene, the direct repeat sequences must be present. So I'm just making a direct repeat sequences. And as you say that this is a target gene, so we cannot place it here, here, or here. We will have to flank the target gene here in one direction, and the target gene has to be flanked in another direction. You look here. This is how the target gene must be flanked. Why? Because the target gene has to be deleted. Now, what has to be done? The target gene has to be deleted. Please do note this particular point. When you have to delete the target gene now, you just have to place the LOXP site or the FRT sequences flanking the target DNA molecule. You must keep FRT sequences just towards the end, upstream, or towards the downstream of the target gene sequence that has to be cleaved. So please look here. Please look here. Pay attention here. This is the short direct repeat sequences that has been flanked. Now the answer.
Correct.
So this becomes correct because I have just taken the target gene, made a short direct repeat sequence, flanked the target gene, and as the target gene has been flanked on both the direction, the target gene will undergo deletion. This is what we have also seen in the case second of the LOX Cree recombinases and in the case of FRT FLP recombinase system. Any doubt, you just clearly ask me. You just clearly provide a message if you have any doubt. Okay. See this solution. When the recombination sites are oriented in a short direct repeat, the result of recombination will cause the deletion of DNA between the homologous recombination site. Note that LOX site should flank the gene and not replace sequence inside the gene. That is why we were flanking the gene on both the sites. Now moving to the next question. Anyone has any doubt about this question? You just provide me a message that's this is my doubt and I will make it clear.
Once the locked sites and Cree recombinase gene have been inserted into the mouse genome,
Now see this. Once this sequence has been inserted and this has been has possessed a short direct repeat. This is a direct repeat. Direct repeat. Okay, this is a direct repeat. And this is a direct repeat which has been inserted. This is a direct repeat. When the two direct repeats are being inserted, then the two direct repeats are undergoing recombination in such a manner that the central present gene of interest, gene will be lost, will be lost. Okay. So this will produce a knockout. How a knockout is being produced? Please have a view.
All of the mice die early in development, before specific tissues have developed.
Now, all the
Question
mice are
3.
going to be dead. So, what does this mean? This means that this gene, which is causing the death of the mice before the mice achieves its maturity, is a housekeeping gene, is a very, very important gene. Okay, and probably a housekeeping gene. Okay, probably a housekeeping gene, probably a very important genes due to which the mice have died. So, question number three.
Which of the following best explains this result?
Now, which of the following is going to best explain this result? Deletion of the target gene causes cancer. Do there is any cancer development? We cannot find any morphological changes, and we cannot see that whether the cancer has been formed internally or not. So we cannot predict the target gene is necessarily for early development. We can say that it might be that the target gene might be necessary for early development, but the results are still inconclusive. There is no positive control and no negative control in the reaction. In the experiment, there are no controls which have been designed, so the results will remain inconclusive. The gene appears, gene although appears to be important, to be important in the embryonic development, but the results are inconclusive as no proper positive and negative controls are available. There are no positive and negative controls available, so you cannot conclude anything out of the reaction. So the results will be the Cree recombination genes disrupt an important gene. Yes, it might have been that they might have disrupted an important gene. Whether the gene was important or whether the gene products were important, we have to just experimentally prove again by taking the positive and the negative controls. So we cannot directly predict the answer, so the results are inconclusive.
Correct.
Okay, so this is what I have tried to explain it to you. Now moving forward,
Que-
before we move forward, I must delete these information. Everything will be provided to you in the recorded version also. You can note from there, just deleting the things. Moving forward.
Question 4. Which of the following experiments should be used as controls?
Now, which of the following experiments should be used as a control in the entire procedure to determine whether the gene was a housekeeping gene or whether the gene was not a housekeeping gene? How can you use the controls? Could we grow the mice under different conditions? No. The growth of the mice under different conditions does not changes the requirement. It will not alter the requirement for that particular gene. It will not alter the requirement, alter the mice requirement, the mice requirement. So the mice requirement will remain the same now. As the requirement remains the same, as the requirement will remain the same. Have some patience, just 10 to 15 minutes more. Insertion of different recombination genes and different recombination sites. So you cannot insert different recombination genes and different recombination sites. You have to check the efficacy of that particular gene. So what has to be done? Insertion of the Cree recombinase at different sites in the genome? No, different sites in the genome does not makes any effect because you didn't know about the size of the gene. So what has to be done? Insertion of lock site but not Cree recombinases. You should insert the lock site but not Cree recombinases in order to find that whether the recombination was taking place by itself or it was a Cree induced recombination. Okay, it could be used in order to determine in order to determine if the recombination was occurring by itself, recombination occurred by itself, occurred by itself, or was induced by itself, or was induced. Okay. Insertion of Cree recombination but not lock site. Okay, again, so do recombination can take place without, do recombination can occur without, recombination can occur without lock site, occur without lock site, without lock site. Both of these experiments will be able to be used as a positive control and a negative control. You know that in the case of the positive control, see this would be serving as a positive control, positive control. In the case of positive control, the cause of a particular phenomena is always there. So there is an insertion of the lock site. You have inserted a lock site. That is why it is a positive control. And here you have deleted the lock site. That is why such type of experiment will be referred to as a negative control. So these are the two controls of the reactions that would be used. Okay. So this, the answer of the above question would be A and B. Any doubt?
Correct.
Now, before I move to the next question, I must delete all the annotations. Deleting all the annotations. Is this clear? Now see this. This is the normal mice. This is the mice plus the lock sites which are present. Mice plus the Cree recombinase. This is the mice in which the Cree recombinase is present, but the lock sites are absent. Okay, you just have to just clarify this question. I would like to clarify this question in front of you. The lock sites are present. Cree recombinase. Cree recombinase are absent. Recombinase is absent. I cannot get the entire word, so I will like write, oh sorry, so I will just write Cree. Okay, Cree is absent. Just a second. Cree is absent. Okay, as Lox is present, so this is a positive control. This is a positive control. Here mice is present, Cree is present. Okay, so this would be Cree is absent, here Cree is present. This would be a negative control because Lox is absent. As Lox is Cree is present, but Lox is absent. Lox is absent. Here was Cree was absent, but Lox was present. Okay, that is why it was referred to as a positive control. This would be referred to as a negative control because Lox is essentially required for the process of recombination. Now here Lox and Cree both are present. The gene X will undergo deletion. Gene X underwent deletion. It is being deleted and the mice has died. Now please do look forward.
While the mice all die early in development as a result of the gene knockout, tests show that the mice do not develop cancer. Apparently, the target gene is important for early development. Question 5. Which of the following tools could be used to determine if knockout of the target gene later in the mouse's life leads to cancer?
Which of the following tool could be used to determine if knockout of the gene later in the mouse leads to cancer? Now, which of the following methods can be used to determine if the knockout of this particular gene is responsible for the occurrence of cancer or not? Again, DNA footprinting assay could be used to predict whether the gene X is a transcription factor or not, whether this the product of this gene will bind to a particular site or not. Okay, so this would be the assay that would be used for to determine if gene X protein will bind to DNA, will bind to DNA. to DNA. Incubation, it does not mean anything, because incubation, you just provide an incubation time period, increase time period for action, time period for action. The next is referred to as tissue specific promoter and transposition. Transpositions, it does not mean anything here, because we know that gene X can't be a transposase. Gene X can't is not a transposase. Is not a transposase. Not a transposase. How can I say that it is not a transposase? Because it would have been a transposase. Nah, sometimes the mice would have died, other time it would have not died, because the gene X would have germed from its particular location, and the lox X site and the lock sites will not have been able to delete that particular gene if it had been a transposase. If this gene would have been a transposase, it would have changed its location from one side to the other, and making the locks sometime functional and sometime non-functional. So the fourth option is absolutely rubbish. It is of no use. Again, incubation cannot be provided. Now, what is given? The only options that finds their importance are the DNA footprinting and tissue specific promoters. Which of the following tool could be used to determine if knockout of the target gene later in the mouse life leads to cancer? Now, this has to be determined by predicting through the tissue specific promoter regions. We know that each and every tissue has a particular promoter. Okay, so if you are going to analyze a particular promoter of a particular tissue, you will find which type of cancer is going to be developed, and whether the gene X is responsible for a development of a particular cancer. Now, cancer can never be holoblastic. That means the entire tissues cannot develop cancer. Only some tissues develops cancer. And so promoter screening would be the best one to screen. Now see this. Cancer is an abnormal normal proliferation of cells developed by specific tissues, developed at a time by specific tissues. So a screening of cells in a tissue specific manner can be used to identify if gene X is causing cancer in a particular tissue or not. Any doubt anyone has, please do raise your answers, raise your queries, please do raise your queries. This is really a typical thing to be understood, because this is for the first time that you have been taught. I do think like that. And if you know that what I'm doing, and if you find any difficulty in any of the answers, please just raise your hand. This would be tissue specific promoters.
Correct.
Okay, so that's all. Now moving to the next question. Before we move, I should delete the annotations made here, making it clear for you all to understand.
Question 6. What can be determined based on these experiments?
Now, what can we determine based on this experiments? Okay. Now see this. There are two experiments which have been added here. Let me just show you what is the experiment. Mice plus B lymphocytes specific Cre recombinase. Mice are going to survive. Mice plus B lymphocyte specific Cre recombinase. Mice plus loxcyte plus B lymphocyte specific Cre recombinase, and then mice is going to die. That means where the recombination is going to take place and where the LOX and the Cre are going to work. Where is the LOX and Cre proteins are going to work. So in this case, there is no death. When there is no death, that means there is the Cree side, but LOX is absent. See, LOX, P is absent. P is absent. So there is no death. But when you put a LOX as well as a CRE, when you put a CRE as well as a LOX, here mouse dies. Mice develop cancer in B lymphocyte. What does this mean? This means that the gene X was the one which was responsible for the development of the cancer in the B lymphocytes. Gene X prevented development of cancer in B lymphocyte, development of cancer in B lymphocytes. Lymphocytes. This is a method that is being used. Okay. So knockout of the target gene causes cancer, at least in certain type of tissues. We always know that. Okay. This is the type of cancer that is going to be produced in certain tissues if the gene is going to be knocked out. The target gene is necessary for early development. Uh, you cannot say that the target gene is always necessary for early development. The Cre recombination causes cancer in B lymphocyte. Yes, the Cre recombinase have the ability to cause cancer in the case of B lymphocytes. So that would be the answer.
Incorrect. You
Yes,
may
please.
want to
Results are conclusive. The Cre recombinase causes cancer in the B lymphocytes. The Cre recombinases. The Cre recombinases along with the LOX are going to cause the cancer. Now,
correct.
okay, so this would be the target was the gene was essentially required in the early development. The knockout of the target gene causes cancer, at least in certain cells, and that one was referred to as the B lymphocytes. Moving forward,
Conservative site-specific recombination is recombination between two defined sequence elements called site-specific recombination sites. There are
so
two families of proteins responsible for site-specific recombination:
this is the conclusion event. Just have a look of what we have studied.
the serine recombinases and the tyrosine recombinases.
Okay, these are the two family of recombinases.
For the serine recombinases, the side chain of a serine residue within the active site attacks a specific phosphodiester bond in the recombination site. The serine recombinases cleave all four strands prior to strand exchange, with one recombinase protein molecule acting on each strand. The DNA strands can be rejoined simply by reversing the cleavage reaction.
This is how the cleavage reaction can take place. Simple, the rejoining of the strands is going to take place, and the cleavage is going to be rejoining the two strands together with each other.
Tyrosine recombinases use a very similar mechanism to that of serine recombinases, except a tyrosine side chain is involved in the cleavage reaction, rather than a serine side chain. The tyrosine recombinases cleave and rejoin two DNA strands first, generating a holiday junction. The other two tyrosine recombinases then cleave and rejoin the other two DNA strands, resolving the holiday junction.
There are two holiday junctions which are being produced in the case of tyrosine recombinases. Cleaves and rejoin two strand first, generates a holiday junction, cleaves and rejoins the two strands, resolving the holiday junction. Here, there is cleaves all four strands prior to the exchange to take place, rejoin the strand by reversing the cleavage reaction, and this is it. Next point, and
If the
the
recombination
outcome,
sites are on
last one.
different circular DNA molecules, the result of site-specific recombination is insertion of one of the molecules into the other.
This will cause the insertion of one DNA molecule into the other DNA molecule.
If both recombination sites are on the same DNA molecule, they can be a direct repeat with respect to one another or inverted with respect to one another. Recombination between a pair of direct repeats will
Cause
result in
deletion,
deletion of the DNA segment between the two sites.
and in the inverted repeat will cause inversion.
In contrast, recombination between a pair of inverted recombination sites will invert the DNA segment.
This has caused the inversion of the DNA segment. Next, this is how serine recombinase work. The four strands have been cleaved. The tyrosine recombinase.
You have completed this
It
exercise.
works, and this is how the outcomes are being produced. This is how we have finished the conservative site specific recombination. We have togetherly finished the Cree locks mechanism of recombination used in molecular biology. We have also finished the flap frat system of recombination. With this, I conclude today's lecture. In tomorrow's lecture, we will continue with the DNA repair mechanism. Thanks a lot for keeping the patience in the entire lecture. Thank you. Thanks a lot. Good night.